A very big hello to my wonderful super champions out there. So how is the Josh still going on? A big salute to all of you who have completed the one-shot video as well as the one marker session, because I can guarantee you whenever you're watching all the videos one by one, you are making sure that you're getting all the marks that you can get from this particular chapter, that is sexual reproduction in flowering plants. Well, this is the last part of the chapter where I am going to discuss the long questions answer. That is, my dear children, if you see, you according to 3 blueprints, we can see there's a 2 marker, there's a 3 marker, and there's a long answer, that is a 5 marker question coming each year. So over here also you can see there's a 2 marker, 3 marker and a long answer, and here in the 3rd blueprint you see a different trend. There's no 2 marker, instead you have 3 MCQs, and then you have at least 1 3 marker and 1 5 marker. So 3 and 5 marker is something that is fixed from this particular chapter. You can expect something around it. And my dear children, how many marks does this particular chapter give you? 11 marks. Principles of inheritance and variation, 13 marks. Molecular basis of inheritance and variation, 13 marks. So 13 plus 13, 26. Yeah, and then over here 11 over here, that means 37 marks though you have got by watching the 3-day session. All of you who did that, kudos to you. And my dear children who are following it regularly, I want to know your names. So put it down in the comment section that, ma'am, yes, we are regular in watching the videos, or at least if you have not watched all the videos, at least if at all you watched this chapter's video, you can let me know that. Okay, so the first question, which is a 2 marker question that has come previously, was put down in the model question paper, was this: complete the tabular column given below with respect to the male gametophyte of angiosperms. So the cells of the male gametophyte, you have to talk about cells, what all are the cells of the male gametophyte, and also the shape of the nucleus of the cells. First of all, what is the male gametophyte? What is the one that is going to give rise to the gametes later on, my dear children? It is none other than the pollen grains out there, right? Now the pollen grains, you know, have 2 cells, one big cell, very big cell, which I call as vegetative cell. So I remember it by V, very big cell. Inside the very big cell's cytoplasm floats another small cell out there, which you call as generative cell. And now this generative cell, how do I remember it? G for gametes. It is the one which is going to undergo mitotic division to give rise to gametes later on. Now both of them basically have their own nucleus also. Vegetative cell, they have an irregularly shaped nucleus, whereas when you talk about generative cell, they have an elongated spindle shaped nucleus. So that is the question. Cells of the male gametophyte, that is vegetative cell and the generative cell. Now shape of the nucleus of the cell, one is irregularly shaped and the other one is what? Elongated. So what would be your answer? Your answer would be this: vegetative, irregularly shaped nucleus; generative cell, elongated or spindle shaped nucleus. And this is the structure. Well, I have spoke about what are the layers: exine, intine. What is exine? Made up of sporopollenin. What is intine? Made up of cellulose and pectin. There is plasmamembrane, yes ma'am. Is there germ pore? Yes ma'am, in the exine. So all of these things we have already discussed, so I am not digging deep, but at least this part of the question should be clear to you. Clear? Done? Understanding? Chalo. Next question. Differentiate between geitonogamy and xenogamy. Both of them are types of pollination. Okay. So if at all, my dear children, if at all there is a plant which has 2 flowers. Okay, this flower has the male part, suppose, and this flower has the female part. From here, from the male part, the pollen grains lands on the stigma of the female flower out there. What do you call this type of pollination? Is this cross pollination or self pollination? First of all, you tell me that. It is cross pollination because it is happening between 2 flowers. But is it true cross pollination? No ma'am. Why do you say it is no ma'am? Well, basically over here, since these 2 flowers are belonging to the same plant, you do not see any genetic variations that are gonna come in the progeny. So which is why I'll say it is not true cross pollination. I'm not crossing between 2 plants. It's just 2 flowers of the same plant itself. Which is why even though it's cross pollination, it is not the true one. Okay, it is not the true one. This type of pollination is known as geitonogamy. Now on the other hand, if you have all the different, totally different plant over here, totally different flower over here, from here to here, pollination is happening. First of all, it's cross pollination, and genetically the progeny which are going to arise, my dear children, will be of what? A different type as well. There will be genetic variation. So if at all you're asked to differentiate between geitonogamy and xenogamy, first of all, it is the transfer of pollen grains from the anther of one flower to the stigma of another flower of the same plant. You first tell the definition. Over here, xenogamy is transfer of pollen grains from the anther to the stigma of a different plant itself. Okay. Now, second point, genetically similar to the parent plant. over here the progeny which will be there will be genetically similar to the parent plant because the flowers are on the same plant itself. Okay, so pollen from the same plant, stigma from the same, now basically the gametes from this, everything is from the same plant itself. On the other hand, genetically different from the parent plant, and which is why it brings variation. Okay, so those are the difference. Now autogamy means, suppose this is a flower, it has both the female and the male part. From here to here it is happening within the same flower, it is known as autogamy. Fine. And that is known as self-pollination. Now next question, what is Parthenocarpy? Give an example. This came in 2023 May. What is Parthenocarpy? Suppose my dear children, over here there is an ovary and inside the ovary there is an ovule. Usually the ovule is the one which after fertilization, after the female gamete over here fuses with the male gamete, this whole thing becomes a zygote, and now what do you call the ovule as? You call this as a seed, and you will call the ovary as what? The fruit. This mostly happens what? After fertilization. But suppose this part has not done anything, there is no fertilization that took place over here, but still the ovary is turning into a fruit without fertilization. This process is known as Parthenocarpy. And now in this case, since there is no change happening up over here, my dear children, do you think a seed will be formed over here? The answer is no. So in Parthenocarpy, there is formation of fruit without fertilization. Second thing, there are no seeds over here. Banana can be made like this, which is why it is one of the seedless variety of fruit out there. Okay, so yes, so if at all, and ma'am, can we do it by ourselves? Can we make the plants produce fruits without fertilization? The answer is yes. So there are certain hormones and the level of the hormones fluctuate, which is why Parthenocarpy happens. Now similarly, if we give the hormones, we induce Parthenocarpy by giving the hormones, my dear children, the same process would be happening, and that is how we give, we produce what? The seedless varieties of fruits. Ma'am, is it good or bad? Depends on what chemicals are being injected over here, mostly considered to be bad, sometimes considered to be good, if at all safe chemicals are being inserted. Okay? Chalo. So ovary develops into fruit without fertilization. Such fruit is known as parthenocarpic fruit. This phenomena is known as Parthenocarpy. Second point will be example. This is how you get your 2 marks. Okay? Fine. If at all you want to show with the diagram, you can show with the diagram also if at all you have time. Now define the following terms: staminate flowers and pistillate flowers. What is stamen? Stamen is the male reproductive part, which is also known as the androecium. It consists of the anther as well as the filament. So the flowers, okay, the flowers which just has the stamen, you call them as what? Staminate flowers. Okay, and now pistillate flowers, pistil is what? Gynoecium, or the female reproductive part. So the flowers which just has the female part, my dear children, you will call this as pistillate flowers. Now ma'am, do we see that usually? Yes, we can see that. You can actually see male plant and female plant. In the case of papaya, they are dioecious in nature. One plant itself will be having all female flowers. Another plant will be having male flowers and male flowers as well. So cross pollination can happen between them. Apart from that, in one plant itself, there can be a case where there is one female flower and one male flower as well. You call these type of plants as monoecious plants because they have both the sexes in one particular plant. Not going very much deep, but just telling you. So dioecious is something that you need to know. Example, the question is being asked. So dioecious example is papaya. Please remember it. I have explained this to you in the one shot as well as the 1 marker session. Please watch them if at all you have not watched it. Staminate flowers, unisexual flower. Unisexual means there is only one sex. So unisexual male flowers bearing only the stamens. Pistillate flowers, unisexual female flowers bearing only what? The pistil. So your answer is this. Okay, going ahead. Sporopollenin is one of the most resistant organic materials known. What is sporopollenin? It is found in the outermost covering of pollen grain. I told you in the pollen grain, my dear children, this is the plasma membrane of the pollen grain, and then comes intine, which is continuous layer. It is made up of 2 things, cellulose as well as pectin. Please remember CP or PC, either way. And now the outermost layer is discontinuous layer. That means it has pores in between. You call these pores as germ pores. Why do you think there are pores? Because over here you do not find something which is known as sporopollenin. If sporopollenin is not there, there will be pores. So everywhere else in the exam you have sporopollenin, which is most toughest material or resistant material out there. If you put alkali, if you put acid, still it will be there, and which is why the pollen grains can be stored in the form of fossils as well. So let's see the answer. So how do you justify this? Sporopollen is one of the most resistant organic materials because it can withstand what? Strong acids as well as alkali. Now, no enzyme that degrades sporopollen is known so far. Because of this property, pollen grains can be preserved as what? Fossils. It can remain for years, my dear children. Sporopollen is found in the exile layer of the pollen grain. Again, you can show a picture properly. It will be really good. Okay? Now, differentiate between pericarp and perisperm, my dear children. What do you mean by pericarp and perisperm? So, pericarp is the wall of ovary. Wall of ovary. Ma'am, have you had mango? Yes, bacha, I have had mango. Ma'am, in mango, the one that you peel off, that is known as epicarp. And ma'am, whatever you eat in the mango, no, it is known as mesocarp. And ma'am, what you throw is known as endocarp. And ma'am, you know what? Inside the endocarp, you have the seed which have its own seed coating that is known as testa and tegment. So these three layers which you see over here, together they are known as what? Pericarp. So pericarp is nothing but the walls of the ovary. Fruit is basically the ovary itself, right? You remember? Fruit is developed from the ovary and the seed is developed from the ovule. So it's the walls of the ovary, that is what is known as pericarp. Now what is perisperm, ma'am? I didn't understand it. So perisperm is quite easy, my dear children. Perisperm is this particular thing. For example, again, ovary is there. You have the ovule. Ovule has its own integuments, inner integument, outer integument, and inside this you have a group of cells which are known as nucellus, okay, which is known as nucellus, which is diploid in nature, not like a normal cell. And inside this one cell will be there which is going to undergo megasporogenesis and later form the embryo sac. So usually the cells of the nucellus, they just go off, degenerate off. But sometimes they persist there. Why? Because they think that the future embryo might need us. Here the embryo is going to be formed now, and the future embryo might need us. We would love to provide it nourishment, so we will stay there. So this layer which is there, which is made up of the nucellar cells, is known as perisperm. Okay, it is known as perisperm, and it is found in black pepper and also beet. So 2B square, you can remember black pepper and beet as B square. These are the things. So how do you differentiate between pericarp and perisperm? Let's see. So my children know the difference now, and now how to write in the form of answers. Come on. Just a second, okay? Come on. Okay. So over here, the wall of the ovary develops into the wall of the fruit and is known as pericarp, and then you have these three layers. Perisperm is what? Persistent nucellus is called perisperm. It is found in what? Black pepper as well as beet. And then you probably show with an example of a diagram. That will be totally fine. On both the sides, whenever you write difference between questions, make sure you are writing like making a line like this and writing on the either sides. Okay, cool. Now we are entering the 3 marker session. Okay, draw, and when we enter the 3 marker session, we see a lot of diagrams up there. Listen, these diagrams are really important from these chapters. Make sure you're listening to me properly and watching the video properly. So draw a LS of grass embryo diagram and label the following part. This is the grass embryo. Grass is what? Basically a monocot, my dear children. It's a monocot. So you are asked about drawing a monocot embryo. As simple as that. Sometimes people say, grass, I have only studied dicot and monocot. So grass come under monocots. So how do you draw this particular diagram? It looks so difficult, right? For me also it was seeming to be too difficult, which is why I went and watched a video, and that's how even I learned. So listen to me carefully. I'm being very honest over here. So make a cap like this, okay? And then make a small hand like this, fine, and then let the body go. And now we are going to make the fins of the fish, 2 fins, and then we'll come back over here, we'll make a small hand. So there is one big hand and one small hand out there. Now my dear children, we will make something like this. Okay, I made something like this, and and I joined it with something like this. So over here also I am making double lines, and it comes like this and it goes to one side. Okay, don't worry, don't get scared. Over here again you make something like this. Okay, here also you make something like that. And now do the labeling, easy. Okay, so I'll tell you how to label it. This thing that you see is known as scutellum. This is one of the cotyledons of the monocot. You know, other cotyledon is not developed, it is rudimentary. This is why it is known as epiblast. Other one got blasted off. It's not even there, epiblast. Okay, it's just there, rudimentary cotyledon out there. Endosperm has not been mentioned in the diagram, so it's okay, you don't have to mention it. Now, my dear children, we all know that is plumule, the shoot apex. You can mention it as the shoot apex. And the shoot apex as well as the leaf primodia, the one that will give rise to the leaf, my dear children, everything is covered by something, very big thing, that is known as coleoptile. So, this is your coleoptile. Okay. Now, over here, my dear children, if this is the shoot apex, then there should be a root apex, which is also known as what? Radicle. Agreed? Now, radicle is covered by something that is known as the root cap. Okay, and all of it together is covered by something that is known as coleorhiza. Coleorhiza. So, R is there, so you can remember R is there for roots, and here P is there for plumule. So, coleoptile is towards the plumule region and coleorhiza is towards this region. Now, you tell me, haven't we marked everything? Yes, scutellum is marked, coleoptile is marked, shoot apex is marked, radicle is marked, root cap is marked, coleorhiza is marked, we marked epiblast also. This picture is perfectly clear to you. Now, you don't have to make this complicated picture, you can just draw this and that will be enough, I think. Okay, cool. So, here what is there? Man with two hands, one small hand, one big hand, and then fins. Okay, two fins are there, and the rest of the thing you have to remember. So, yeah. Now, going ahead, draw labeled. If at all you think that that idea was nice, definitely let me know. Okay, and draw a label diagram or transverse section of young anther, my dear children. Transverse section of young anther, that is, you're cutting like this, the anther like this, and you're looking from the top. How does it look? So, you have to draw that structure. This is the structure, but you have to draw it properly. Okay, so how do you show it? So, again, this is the anther. Draw it properly, okay? As of now, I'm not drawing that great. So, over here you have this sporogenous tissue. Okay, this is what? The sporogenous tissue. Now, just outside the sporogenous tissue, you have cells which are known as taptal cells, and together you call them tapetum. Now, we directly go outside. Okay, so over here, these cells up over here are known as epidermis. Make it properly, label at one side, okay, and then after epidermis comes the next layer, which is known as endodermis, and within endodermis, okay, within endodermis and after tapetum, in between both of them you have another layer which you call as, which is in the middle. So, you call this as, easy name, no, middle layer. You call this middle layer. So, you have to make at least one part like this and just show. That would be enough. Rest of the parts you can just make round things like that. Cool? Works? Fine. Now, next question. Write any 4 characteristics of flower pollinated by wind. Wind pollinated flowers. First of all, tell me. The pollen grains which are there of these flowers, my dear children, are they sticky or not? They are non-sticky, ma'am. That's how they can get to fly. So, non-sticky pollen grains. Now, pollen grains are present on what? The stamen. The stamen, how should be they? How should they be? They should be well exposed so that the wind can come and take the pollen grains. So, well exposed stamens should be there. Now, my dear children, these pollen grains are going where? The pollen grains are going where? They are going to the stigma. How should the stigma be? It should be, tell me, it should be sticky. The stigma obviously is sticky. Apart from that, it should be feathery in nature. The stigma which is there, my dear children, or not the stigma exactly, the whole, what do you call, the pistil itself, it should be feathery in nature such that, okay, such that, my dear children, it can capture the pollen grains. 3 things are done, and apart from that, I'll tell you lot of other points over here itself. So first thing, wind pollination requires the pollen grains are light also, yeah, light in weight and non-sticky and powdery, so that they can be transported easily. They often possess well exposed stamens, I told you, and also feathery stigma to easily trap airborne pollen grains, and must have, usually the flowers which are there have a single ovule. If you have the flowers which are there, they don't have multiple ovules, my dear children, they have one ovule. Why? Because wind pollination to happen is kind of, what do you call, it's not a paka event. It can happen, it may not happen. So which is why you do not want to waste your plant's energy in making multiple ovules out there. So usually the wind pollinated flowers will have a single ovule itself, and now numerous flowers would be packed into inflorescence, just like how it is happening in the case of insect pollinated flowers. There the numerous flowers, multiple flowers, if at all they are there, they'll come together to form what? Inflorescence. So that, my dear children, at least some stigma can capture it. Okay, example of wind pollinated, you have two examples, please remember both of them. One is grass wind pollination is very common, and the second one that you have is this thing, corncob. And in corncob, if at all they show you the picture of corncob with tassels, with the, what do you call, thing that we tear off and throw, they are nothing but the stigma and the style of the corncob, my dear children. Now the next question comes, what are false fruits? Gives an example of it. False fruit means basically, what is a fruit? Proper fruit. From the ovary, if you get a fruit, that is known as fruit. True fruit. If at all, apart from the ovary, some other things are also participating in the formation of the fruit, my dear children, this will be known as false fruit. Suppose, so there is the ovary resting on the thalamus. So thalamus is also now coming and forming a part of the fruit. So what will this fruit be known as? This will be known as false fruit. So false fruit develop from parts other than the ovary, like the thalamus or the pedicel as well. This part is pedicel and this is the thalamus. Example, you have apple. In apple, you saw the thalamus is also taking part in the formation of the fruit. In cashew also, from the pedicel things can come. Strawberry also is an example. So apple, cashew, strawberry are examples of this. Please remember it, very, very important. This picture we discussed in the previous sessions. So please watch those videos as well. Okay? Now describe the structure of dicot embryo with the help of label diagram. Again a 3-marker question, again a diagram. How do you remember a dicot embryo? First of all, tell me the example of dicots. Any one of you who remember the examples of dicots, put it down in the comment section. I'll tell you one example, groundnut. The ones that I can break into two, which have two cotyledons, are known as what? Dicots, dicot. So give the examples of dicots other than groundnut. So over here, my dear children, easy understanding, all the embryo has an embryonal axis like this. Okay. The upper part will be having plumule at the tip, the lower part will be having radicle at the tip. And the radicle is mostly covered by something which you call as root cap, because it has to go into the soil and it should not be damaged. Now my dear children, cotyledon is there. So one cotyledon goes like this. This is one cotyledon, and this thing that you see over here is the next cotyledon that you have. So dicots, two cotyledons are there over here. Now this part, the part above the joining of cotyledons, this is known as epicotyl, and this part below is known as hypocotyl. Below what? Below the region of attachment of cotyledons. So from here it attached, right? So below it you have hypocotyl and above it you have epicotyl. In the epicotyl region you have the plumules, and these things are nothing else but the cotyledons. Cool. So in the picture they have not properly mentioned the plumule, epicotyl, but have told you, so please remember it. So if at all, describe the structure with the help of a label diagram. So obviously don't just put the diagram there and come, put at least 1 or 2 points out there as well. So it consists of an embryonal axis and 2 cotyledons. The portion of the embryonal axis above the level of cotyledons is the epicotyl, which terminates with the plumule or the stem tip. The cylindrical portion below the level of cotyledons is hypocotyl, that terminates at the lower end at the radicle or the root cap. That's all. And the root tip. So radicle or the root tip, and is covered by what? The root cap. That's all. Easy. Right? Easy 3 marks. Now, this is not 4-marker question. This is the 5-marker question. Okay. So, picture related to pollination is given below. This is the picture. Will this pollination confirm fertilization? What do you think? There is an insect sitting on this. Do you think this pollination will anyway confirm fertilization as well? What is pollination? Pollination is transfer of pollen grains from the stamen to the stigma, right? What is fertilization? Fertilization is fusion of the male and the female gametes. So, obviously this has to happen for fertilization to happen, but if this has happened, pollination has happened, do you think fertilization will anyway happen? No. There are chances that it may not happen. Pollen tube formation does not happen. Pollen tube does not reach over there. The male gametes do not reach over there. What will that case be? My dear children, in that case, fertilization would not be happening, right? So, will this pollination confirm fertilization? Maybe, maybe not. How do you frame the answer? Let's see. No, pollination not always confirm fertilization. Pollination is what? The transfer of pollen grains from the anther to the stigma, whereas fertilization occurs if the pollen successfully germinates from the pollen tube and reaches the ovule to what? To fuse with the egg cell. So, there is no guarantee that every pollination can end up in fertilization. Now, what are the floral rewards provided by the plants to the insects to revisit? Now, this guy came over here to this particular plant, and it took the pollen grains and it helped in pollination to happen. But what does the plant do in such a way that the bee comes again the next time? What are the floral rewards that it provides, my dear children? Usually nectar, right? Nectar. And also one more thing, that is the pollen grain itself. So, pollen grain itself is very nutritious, that is, so it's not like, so you might be thinking, ma'am, if it takes the pollen grain, then how will pollination happen? So, obviously it will eat some of it, it will take some of it, not all of it, right? So, some of it will be still kept there, some of it will be still transferred. So, what are the pollen rewards that you get, my dear children? First thing, as I said, nectar as a food source, pollen grains as a source of proteins, and apart from that, if you just not talk about bees, if you talk about moth and some other organisms, sometimes plants also provide a shelter. Shelter for what? For laying the eggs. So, within the ovary they provide space for it to lay the eggs as well. So, shelter, safe places for laying eggs. Example, yucca and moth interaction. This is one important example, no? Yucca and moth. I've taught you that. So, please remember all these things. Yeah? Now, mention the characteristics of flowers in this pollination other than the floral rewards. Floral rewards, you forget about that. Other than that, what will be the other characteristics of these type of flowers? Whereas insect pollinated flowers, my dear children, they would be colorful, they would be fragrant. Okay, they would be colorful, they would be fragrant if it also bee and things like that. Sometimes if it also is a fly, it would have a foul odor to attract them, because flies not like the good odor, they like the bad odor. Right, apart from that, my dear children, the small flowers would be coming up together to form what? Inflorescence. They would be clustered together to form inflorescence. Right. Now, what about the pollen grains? Over here, when pollination we studied, they should be non-sticky, but over here they should be sticky so that they can stick on the surface of the pollinators. So, all these features are something that you need to remember. So, majority of insect pollinated flowers are large, they are colorful, fragrant and rich in nectar. Small flowers, they cluster together into form an inflorescence, and animal attraction by the color and the fragrance. Flower pollinated by flies and beetles, as I told you, foul odor to attract the animals. And one very important thing which is not being mentioned over here, the pollen grain should be sticky. Please remember. Okay, done? Got your 5 marks? Now, explain the structure of Megasporangium. So, I would love to draw it and explain to you, because that's how I think you guys will remember it better. We have drawn it several times, but again. So, over here I have the ovary. In the ovary I have the ovule. The ovule is feeling very cold, so it has 2 sweaters. One sweater is this, that is one integument, inner integument, and then there is one more sweater. This is known as outer integument. Ma'am, why have you not buttoned it up? Because there is a small pore over here, and this pore is known as micropyle. And these integuments, I'll just write it, they are integuments. Now, where is this all happening? Inside the ovarian cavity. This is the cavity of ovary, and this is also known as locule. Now, ma'am, is there something else over here? Yes, Baba. There is what? There is placenta, which is there for the nourishment of the ovule. So, ma'am, the placenta is there, ovule is here. What is that? Something that would be connecting them. So, over here they have something that is known as funicle. Okay, there is funicle which is going to connect what? The placenta to the ovule. And what do you call this connection spot as, ma'am? Do you have a specific name for it? Yes, Baba, and it is known as what? Hilum. It is known as hilum. Ma'am, you said that this is ovule, right? Yes, I said that this is ovule. What is inside the ovule? Inside the ovule you have so many cells, Baba. And what do you call these cells as? What do you call the cells? You call the cells as cells of nucellus. It's not nucleus, it's cells of nucellus. One such cell, okay, which is known as the megaspore mother cell. the megaspore mother cell is going to undergo meiotic division to form the whole functional megaspore, and later it forms the embryo sac. Okay, so these are the things that you need to remember. Also one more thing, if this is the micropyle end, the other end will be known as the chalazal end. This will be known as chalazal end. Now my dear children, somewhere, sometimes the placenta can be here, funicle can be here towards this region. Does not mean chalaza also move towards this region. Wherever you have the micropyle, opposite end of it will be chalazal end. Always remember, if at all you are asked where is the egg apparatus, or where do you think the division happens of the megaspore mother cell, everything happens more towards the micropyle region only. In the chalazal end, always remember, in the embryo sac you have certain cells known as antipodal cells, they are there in the chalazal end. That's all. So yeah, take a picture of it if at all you want, or else let's go back to this. So now, the structure of Megasporangium. The ovule is a small structure attached to the placenta by the means of a stalk called, I told you, it's known as funicle. The body of the ovule fuses with the funicle at the region known as hilum. So the point of attachment is hilum. And the ovule has one or two protective layers which are known as what? Integuments. These encircle the ovule except at the tip, the small opening which is known as micropyle. Now the chalaza is present opposite to the micropylar end. Okay, the integuments enclose a mass of cells with reserved food material. Now what do you call these mass of cells as? Nucellus. Nucellus encloses the embryo sac or the female gametophyte. Now whenever a female gametophyte is formed, it would be inside the nucellus, The cells of the nucellus is usually going to be used up, sometimes it persists, and that's when you call this as perisperm. Okay, now you have the 13th question. Do we have more questions after this? I think there is one more question. Yes, one more question. Okay, the picture of mature angiosperm embryo sac is given below, and answer the questions that follows. Okay, so which cells, nuclei of the embryo sac produce zygote and the primary endosperm nucleus? So what do you think, which of them are going to produce? So my dear children, over here you have the egg cell. This is the egg cell which is having n number of chromosome. This is going to fuse with the male gamete, which is also going to have n number of chromosome. Together they are going to form what? Zygote. N plus n, it becomes what? 2n. That means it is diploid in nature. Okay, agreed? Which cells, nuclei of the embryo sac produces zygote? So you know it is egg cell. Okay, and the male gamete which comes and fuses with it. Next thing, which is the one that forms what? The primary endosperm nucleus. So let's see. There are always two male gametes. So one male gamete came and fused over here. The second male gamete, my dear children, the second male gamete, which is having n number of chromosome, fuses with the two polar nuclei. So two polar nuclei, two of them both having n. So in total how many n is there? 1, 2, 3. So 3n number of chromosomes are going to be formed in the new nucleus, which is known as the primary endosperm nucleus, right? Primary endosperm nucleus. Now this nucleus belongs to which cell? Central cell? Now you don't call it central cell, you will call this as primary endosperm cell, PEC. Okay, it is triploid in nature, always remember. Now what is the ploidy of antipodal cells and the primary endosperm nucleus? So primary endosperm nucleus, you know it is triploid. What about the antipodal cells? Antipodal cells are these cells. And all of them, as usual, all of them initially had n only. So they are also going to be haploid, my dear children. Don't think all of them, let's plus n plus n plus n, don't do that. Talking about individual antipodal cells, it will be haploid in nature. Half the number of chromosomes will be there. In all of them, half the number of chromosomes are there in all the nuclei. But if you talk about the cell, the central cell, since it has two polar nuclei, so n plus n, 2n. That is the only diploid cell, and now it has become triploid also. Okay. Now, what is the ploidy of antipodal cells and the primary endosperm nucleus? The antipodal cell, as you know, it is haploid. If you talk about primary endosperm nucleus, it is what? Triploid in nature. Now, ma'am, why the endosperm development precedes the embryo development? Why do you think endosperm develops first and then the embryo? Ma'am, zygote is being formed. This zygote will transform into embryo, right? So before that, why do you think endosperm is needed? So the answer to this question is that you want to transform the zygote to embryo, no? Which is why you need nourishment source, and that nourishment source is what? Endosperm. This is why endosperm is the one that should be forming first. Don't think that endosperm is formed now. No, it will undergo division to form a proper endosperm tissue later on, my dear children. And then only the zygote will transform into what? A embryo. And how do you frame answers for all this question? That is important. So over here, which cells or nuclei of the embryo sac produces zygote and the primary endosperm nucleus? The egg cell haploid fuses with one sperm nucleus or one male gamete nucleus to form what? Zygote diploid 2n. Okay, the two polar nuclei, each haploid fuses with another sperm nucleus to form primary endosperm nucleus PN, that is triploid. Now what is the ploidy of the following? Antipodal cells, they are haploid. Primary endosperm nucleus, they are triploid. Why does endosperm development precedes the embryo development? As I told you, the endosperm acts as what? Nutritive tissue and provides essential nutrients to what? Developing embryo, which is why the early endosperm formation ensures a steady supply of food for proper embryo growth as well as development. Khatam. Okay. So please write your answers like this. Now coming on to the last question that we are having. Explain any 3 outbreeding devices in flowering plants. What do you mean by outbreeding devices? So my dear children, whenever a flower basically or a plant has a condition like this, male and female. Suppose this and this, for pollination, fertilization, everything happened. What's the problem with this is that you are basically forming the progeny which is of the same type. And this causes something that is known as inbreeding depression. Plants also get into depression. So inbreeding depression means basically, suppose these plants, this parent plants had toxic character, that is, it was not resistant to anything, it is not pest resistant, not drought resistant, nothing good is there, and now the progeny is getting it again and again. Do you think the plants can survive the progeny once? No. Which is why the nature itself do not want this to happen. Okay, they do not want this to happen. They want proper cross pollination to happen. So there are certain outbreeding devices, okay, which allows cross pollination to happen, my dear children. So what are those devices? Some devices are there in the plants genetically, some, yeah, it happens like that. So one such device is male and female flowers are present in different plants. Okay, male and female flowers which are there, they are going to be present in different plants. So dioecious plants, as I told you. So here it will be having the female plants, and over here it will be having the male plants. So obviously in this case, from fertilization or what do you call as pollination can happen like this only, cross pollination can only happen. And so this is the case of dioecious plant, example papaya. So male and female flowers on different plants. Okay, unisexual male and female flowers on different plants. Or unisexual flowers, if both male and female flowers are present on the same plant, such as castor and maize, monoecious, it prevents autogamy but not geitonogamy. That can be also possible. So this is dioecious case where unisexual plant itself is there. Yeah, unisexual flowers can be there, as in there can be a female flower, and in the same plant there can be a male flower. So to some extent it is preventing self-pollination. Self-pollination is definitely prevented. Autogamy is not happening. But geitonogamy can still happen. Geitonogamy can still happen. What do you call these type of plants as? You call these type of plants as monoecious plants, which have both the sexes in the same plant. Okay, such as castor and maize. So here self-pollination is prevented, but cross is not what you call completely true cross. So over here geitonogamy is happening, but in this case, in the case of dioecious plants, xenogamy is happening, but still they can be considered. So unisexual flowers, if both male and the female flowers are present on the same plant, such as castor and maize. Monoecious, it prevents autogamy but not geitonogamy. Please remember, question can be framed in a different way, can say that autogamy is happening but not xenogamy. It's not like that. Xenogamy can still happen between this plant and some other plant. Geitonogamy is something that is happening over here. Autogamy is something that is not happening over here. Now next is that in some species, pollen release and the stigma receptivity, you don't need to write dicogamy and things like that. You can just directly write, okay. In some plants, pollen release and stigma receptivity are not synchronized. The pollen is going to be released, but the stigma is not going to be receptive. And if the stigma is not receptive, even though the pollen comes and lands, my dear children, it won't be able to germinate. So this is not being synchronized. And this will obviously prevent what? Self-pollination, and favors what? Cross-pollination as well. Now the next point is flower in which anther mature earlier to stigma to prevent self-pollination. There are certain flowers in which what? The anther is going to mature first and then the stigma. So in that case also the timing gap is there. Okay. Now next is what? Flower in which stigma matures earlier to the anther. That is also there. Now one more thing is there, self-sterility or self-incompatibility. So this is totally a genetic mechanism. The pollen grains that do not, so pollen grains, even though they land on the stigma, my dear children, the plant will not allow germination to happen. This is known as self-incompatibility. This is a genetic mechanism, you need to remember it. Self-sterility or self-incompatibility. So what already study? Unisexual flowers or unisexual plants, my dear children, both of them. And then you saw that the timings, the time synchrony is not there. Pollen release and stigma receptivity, it's not synchronized. And in some cases, the anther matures early than the stigma. In some cases, the stigma matures early than the anther. Okay, and then self-incompatibility, which is genetic mechanism to prevent what you call fertilization. And there's one more thing, that is difference in the locations, my dear children. Suppose over here the anther is located like this and the stigma is at the top. Don't you think it's very difficult for the pollen grains to go up like that? Okay, even through wind and everything. So yes, that is also one very important thing. So that also prevent what do you call pollination, self-pollination up there. Okay, please remember monoecious and dioecious, very, very important. Now, one more thing is there, you might be asked this question which came the previous year actually, which is why I was very curious to tell you guys, and that is, what is that method which the farmers use to produce superior variety of crops? And that method is known as artificial hybridization. So, artificially you are going to hybridize. How, my dear children? Suppose this is a plant. Over here I have the female and the male part. So, if at all it's a bisexual flower, bisexual means when it has both the sexes. If at all it's a bisexual flower, I am going to remove the anthers because I do not want them to contribute their pollen grains. This process is known as emasculation. Emasculation. This is only needed if the flower is bisexual. If it is unisexual, it is not needed. The second step that you need to do is that you have to cover this stigma or the whole thing. Why? Because you don't want any pollen grains to come over here. And also you want to wait. You want to wait such that it becomes receptive. Now this is known as bagging. Now after bagging, my dear children, you will remove the bag. Whenever stigma is receptive, you will remove the bag and you will put the desired pollen grains out here. For example, I took a plant who is really good with what? Resistance to pests. So I took that plant's pollen grains and I put it over here. I have to look for the species and everything that is there. But anyway, I did that. So the third one will be what? I will provide what? Pollen grains from the desired plants. And then, my dear children, again I don't want any other pollen grains to come over here, so I will do the fourth thing, which is what known as rebagging. And together all these processes, you call this as artificial hybridization. Artificially you are going to produce hybrids. That is why it is known as artificial hybridization. Done, dusted, and that's all. I don't want to take any bit of your time now. You can go, you are free. You got 11 on 11 on this particular chapter. You can just assume that and you can go. And definitely, if at all you have any doubts, put it down in the comment section. I am here to help you. And if at all you have not subscribed to the channel yet, subscribe to it right away to not regret later on. Yeah, that's all. Bye-bye. Take care. Lots of love.